So we're very, very honored to be here today. I can't tell you how much. My name is Tybjærg-Hansen. I'm a professor of clinical biochemistry and translational molecular cardiology at Copenhagen University and the chief physician at Copenhagen University Hospital. It's called Rigshospitalet, and it's the ugly one to the left.
Yeah, my hospital, the other one. So I'm equally really honored also. I think I like to say that we put a title from our talk. We call it FH, LP-little-a, and LP-little-a and FH. These are two areas we have written about a lot and given talks about to contribute. Not saying that there's not a lot of other scientists in the world that have contributed as much or even more than we have, but this is our talk, so we talk about what we have been publishing.
Yeah, so my early scientific training started in Copenhagen at the lipid clinic at Rigshospitalet with a person who is now deceased, Hans Meinertz. And then I moved to Hagen Research Lab, which is now under Novo Nordisk, and I worked with Jørgen Nierup, who's a diabetologist. But by far the most important person for my scientific career was or is Steve Humphries, Professor Steve Humphries in the UK, and I worked with him for 2.5 years while in London.
So about the same time, I did a parallel travel, you can say, from Copenhagen, where I started my training, Steen Stender, and then I went to New York, Cornell University, upstate New York, with Donald Silversmith for 2 years, and then from there to London, where I worked with Barry Lewis, and actually there we lived together. We dated long distance across the Atlantic for a couple of years, and then we lived together in London. Early work.
Yeah, so here's some early scientific work, genotyping for FH. This is how old I am. It's a southern blot, and it's detecting ApoB3527, or familial defective ApoB100, it's also called. And you can see we have 8 rows, and that's 8 patients, and patient 1, 6, 7, and 8, they are heterozygotes. They have 2 alleles, an A and a G allele. So they have ApoB3500 or 3527. This is not something you want to do on hundreds of patients, I can tell you.
Yeah. My early work was a bit different. I actually started with rapid work, kinetics, and also some human studies. And some of my early work was trying to understand how lipoproteins in plasma interact with the arterial intima where atherosclerosis occurs, leading to coronary heart disease and other atherosclerotic cardiovascular disease. And what I showed was that LDL and remnants, they can come into the arterial wall and get trapped there and cause atherosclerosis, while the very large one, chylomicrons, that have a diameter more than 75 nanometers, they were simply too large to come into the arterial wall.
Yeah, this is from when we first met or about there, and very unscientific. As you can see, Borg, he had more hair, and he also looked like a mafioso.
Next slide, please.
Yeah. So these are the large human cohorts that we have collected over time, and they have been very important for our scientific work. So the first and oldest and also smallest is the Copenhagen City Heart Study, which started in 1976, actually started by my father. And it included at that time 15,000 individuals. And in the third examination in 1991 to 94, we collected DNA on 10,500 individuals, and people actually thought I had gone mad. We now have 47 years of follow-up.
Yeah, we later realized that this was actually too small from the future, and therefore I started this other one in 2003 called the Copenhagen General Population Study, which we now have 160,000 recruited. And I think the beautiful thing about doing these studies in Denmark is that we have all these registers, so we have 100% complete follow-up. We don't lose track of a single person. We know exactly when they die or if they immigrate from Denmark.
But let's move on to familial hypercholesterolemia. So to the left here in red, you have some original papers on FH from the group, starting with a paper in New England Journal of Medicine in 1998, and then some other papers. I have to say a person who has been very instrumental in this research is Marianne Bin.
And then we were very fortunate to be invited to be part of consensus paper from European Atherosclerosis Society's chairman by John Chapman and Henry Ginsberg we have here today. And Henry and John, they were fantastic getting started with these things. 3 consensus papers, and down below one from the Family Heart Foundation, back then FH Foundation, that was also involved in. That was great papers, I think.
But FH is, of course, a clinical diagnosis, as you'll know, and in about 40 to 50%, depending on the referral criteria, you will find mutations, and the mutations will be in the LDL receptor or in apolipoprotein B, as you heard, and these kind of mutations, they inhibit the uptake of LDL cholesterol in the liver, or LDL in the liver, while PCSK9, which is a rare cause, targets the LDL receptor for degradation. Either way, it'll result in an elevated LDL cholesterol in plasma, in premature atherosclerosis in the arteries, and in premature coronary heart disease, myocardial infarction, and angina pectoris. Now, the most common genetic cause is mutations in the LDL receptor, but actually the second cause, as you'll also hear later, is lipoprotein a, and that's because lipoprotein a is almost entirely genetically determined. Then mutations in ApoB come third, and PCSK9 fourth.
So here's some recent data, meta-analysis we published in 2020. And I think to show this one here, the main point is really all the gray countries in the world, there's absolutely no idea how common is FH is there. Nobody's screened for FH in these countries, and that's by far the most countries in the world. And the ones with colors, these are the ones where there are publications about how common it is. And you see some of the very dark ones are the ones sometimes with founder effects, or in Africa, because there's a lot, but particular founder effects. I think the summary of this meta-analysis, which was published, you could say, almost back to back with another one in Circulation, but this is our paper. We came to the same results, really, that in the general population, roughly 1 in 313 have FH, and in those with ischemic heart disease or coronary heart disease, 1 in 31. And for those with premature ischemic heart disease, 1 in 15 had FH. And if you look in studies of people with hyperlipidemia, it was 1 in 6 with FH.
And here's the cumulative LDL cholesterol in millimoles per liter. So this is what you will eventually accumulate also in your arterial wall. And it's a function of age. So the older you are, the more you will accumulate. If you don't have FH, that's the green curve, then you'll reach the threshold of about 160 nanomoles per liter, and this is where you get clinical symptoms at the age of around 55 perhaps. However, if you're heterozygous for FH, you'll reach this threshold before that, maybe at 35 years of age. But if you start treatment at low dose, for example, of statin at an early age, already in childhood, or start later but with a higher dose, then you can bend the curve. That's the turquoise and purple curves here. And of course, if you're a homozygote, you'll reach this threshold already perhaps when you're around 12, at least when you're below 20. And that means that it's important to treat this disease because you can bend the curves.
And so I have the pleasure to show you some unpublished data that we'll, I think, submit maybe next week or week after. It's this opportunity in Denmark that I realized that we can actually study FH in Denmark, the entire population from 1978 through 2018. And this is because, as shown in green, that WHO, International Classification of Diseases, ICD system, they never had a code for FH. And we know here from your foundation actually getting one from the US, but it turns out that in Denmark, somebody was smart enough to make a modified version of FH, so it has been registered in Denmark since 1977, which is unique. I don't know of any other country. So we took the Danish civil registration system that each of us have a number like everybody else living in Denmark, and then we looked at all-cause mortality and cardiovascular disease in the health registries from 1978 to 2018. And here's bottom line results is that on this age scale from the 70s until close to today, what was the age at death of the people that were giving a diagnosis of FH compared to the ones that were not in the entire population of our little country, Denmark? And you can see in the 70s, there was 20 years difference. The ones with FH, they died 20 years before the other ones. But this difference has narrowed together, so today, there's actually no difference in the age at which people die. We ascribe this to the much better treatment of people with acute coronary syndrome. In Denmark, it's fantastic. If you get acute coronary syndrome, you get into hospital immediately, you get a PCI. So we keep people alive. I should say, though, when we look at the next thing, this is the same people will now look at what is the age at which they get a diagnosis of acute coronary syndrome. And back in the 70s, there was 19 years difference between those with and without FH. And today, it's still 10 years. So there's ample reason for let's find everybody with FH, even in our country and even in countries with very advanced healthcare system where they keep people alive because they do get coronary heart disease and whatever you get from that, heart failure, for example.
But let's move on to lipoprotein little a. So lipoprotein little a, here's the number on the y-axis, the number of papers on lipoprotein little a, and as a function of the year on the x-axis. LP little a was actually discovered already in the early 1960s by a Norwegian physician called Børge. But you can see there are not many publications at that time on LP little a. Another very important person with regard to LP little a was Gerd Uttermann, an Austrian physician. However, it wasn't until the 1980s when Lauren and Skenew in the States cloned the LPA gene that the interest in LPA really started to get momentum, as you can see by the rise on the curve here. But what happened then was, unfortunately, some very prominent researchers from the US published 3 papers in JAMA which concluded that LPA was not associated with cardiovascular risk. And this led to, as you can see further on to the right, simply a loss in interest in publishing on LPA until, as Florian Kronenberg pointed out, LPA was resurrected by genetics. And following that, and 3 rather important papers using genetics here in 2009, there was an increase in interest in LPA again, and of course now we are eagerly waiting the results of the phase 3 trials.
I think an important comment, Anne, was that these 3 papers published in JAMA was actually because they used the wrong assay and bad samples, so that's why. So with 40 later on, it was shown that LP little a in these studies were actually describing who would get coronary disease later. So we ourselves also work with genetics, and this is our son Ask and our daughter Liv, and when you look at the son, there's no doubt where he comes from, right? I mean, it's looks a bit like the mother, but the daughter, you could really, where does she come from, you know? But here's the answer, you know, so.
Yeah, okay, so it wasn't the postman, Borg. But as you can see, she's wearing a little white there, and if you go back on the slide, he is also the son wearing a little black there.
I think telling me it's not only genetic, but there's some environment also involved here.
To the left again, we have some original papers, the most important ones on lipoprotein little a, and I think by far the most important of these was the 2009 paper in JAMA, and the first author is Pia Kamstrup on that paper.
And also here we've been involved in consensus paper, the first LP little a, and the low one, the one Florian Kronenberg just wrote a year ago, and also one together with the National Lipid Association, Don Wilson, first author.
Yeah, and here's the data from the 2009 paper. It uses Mendelian randomization to prove causality of lipoprotein a on risk of myocardial infarction. So what it shows is risk of myocardial infarction for doubling of lipoprotein a levels in an observational study at the top, so simply using plasma lipoprotein a, and at the bottom using genetically determined lipoprotein a levels. Now if there's no increase in risk, then you have what's called a hazard ratio of 1, but if you're to the left of 1, then you have an increased risk. And there's also overlap between the observational and genetic estimates, signifying that there's causality here between higher lipoprotein a levels and risk of myocardial infarction.
Later come this paper from George Tanasulis from Canada, published in New England Journal of Medicine. This is what's called a Manhattan plot or GWAS study. On the x-axis is all the different chromosomes, and all the single dots in blue or green are single genetic variants. And on the y-axis you see how significant they are related, in this case to aortic valve calcium. And the one that came out by far the most was an LP little a SNP. The same one we knew for coronary heart disease. And in that paper, it was also shown to be associated with a aortic valve stenosis.
And then Pia Kamstrup showed this observationally, namely that the higher the LP a levels, the higher the risk of aortic valve stenosis.
Here's sort of like summary on all our genetic studies from our Copenhagen cohorts. What is the genetic evidence that LP is a causal factor for these diseases? And what we find in our studies is that for aortic or atherosclerotic stenosis, it could be in the carotid arteries, in the coronaries, or in the femoral arteries, up to 1.5 to 5-fold increased risk. For myocardial infarction, 1.5 to 3-fold. For aortic valve stenosis, 1.5 to 3-fold. And this condition that comes after myocardial infarction and aortic valve stenosis, namely heart failure, showed by these feet with water in them, 1.5 to 1.7-fold increased risk. Ischemic stroke, 1.6-fold, and all-cause mortality, 1.2-fold. That means 20% higher risk of dying early on than if you do not have it.
Yeah, and these increases in risk, as Børge pointed out, for atherosclerotic stenosis and for myocardial infarction, aortic stenosis, and heart failure, they compare the top 33% of LPA levels with the rest, whereas for ischemic stroke and all-cause mortality, it's even more extreme. It's the top 5% versus the rest.
So, LP(a) in familial hypercholesterolemia.
Again, we have some original papers that are important to the left. The most important one of these is the top paper, first author Anne Langsted.
And there's these 3 consensus papers we already mentioned. Some of the LP stories mentioned there, but of course there's not a consensus paper directly addressing this issue. What we did find in that Lancet Diabetes Endocrinology paper was that after screening 46,000 individuals from the Copenhagen general population, we found 1 in 200 had clinical FH. And when we ranked the causes for clinical FH, LDL receptor was the most important, but high LP(a) could explain 25% of everybody getting a clinical diagnosis of FH. And then ApoB and PCSK9 mutations explain a little also.
So here you should look at the top of the slide. So what you see to the left is individuals with FH, no for the 2 first ones and yes for the 2 next ones. And then to the right of that, the lipoprotein a level at or below 50 milligrams per deciliter or above 50 milligrams per deciliter. And if you look at the brown spots and the red spots, in brown, individuals with FH, they have an increased risk of myocardial infarction, of course, but if you add a high lipoprotein a, your risk increases even more. So very high risk here. And if you use genetics, which done at the bottom, then you see the same increased risk with increasing lipoprotein a levels.
So this is just how I look upon the evolution of genetics understanding of FH. So down below, this is what it used to be when I went in medical school. FH was basically a clinical diagnosis. And then came the LDL receptor research from Brown and Goldstein. I got the Nobel Prize. It took some years, that's the sort of like yellow arrow, that took some years before it really, people in clinical working with FH realized that LDL receptor was actually important, and we should probably maybe think about testing about it. Later came ApoB, LDL, RAB1, and PCSK9. It's the same, it took some years from the research until it came into clinical practice, thinking about this as a cause of clinical FH. And now we have the same situation with Lp(a). It's there, 25% of all clinical FH. I think it'll be a few years before all of you also will say that, well, this is actually the 5th gene causing familial hypercholesterolemia. These three are dominant. This one is recessive, and Lp(a) you could say is a multi-allelic version of genetic causes of clinical FH.
So what's in the future for Denmark with regard to FH?
Yeah, and implementation, you can say. So I've been involved in setting up this Danish quality control database for FH, including testing for Lp(a). I talked with a lot of politicians, and we ended convincing them to fund it. So it's completely paid by the Danish taxpayer. So when we started in 2020, got it established, and Denmark has these quality control databases paid by taxpayers. There's 80 of them now, and I think this was the number 80 actually. We have a plan for 10 years. We'll find everybody with FH. So around 2030, a little bit later, we'll have found the 30,000 with FH in Denmark. And we have these quality control things, and this is one quality control. There's a total of 14 that we as scientists and clinicians could tell, and the government accepted it, and that's it then. So here's 2 other ones. So we said that at least 70% of everybody suspected of FH should have a genetic test, and at least 80% of those suspected of FH should have an Lp(a) test. And once a year, there's a report from this government agency, and it's been sent to all the hospitals in Denmark, and they are told, how is your hospital doing for these 14 quality control for Lp(a). If they're not doing well, they have to explain back, why are we not doing so well as other hospitals in Denmark.
Yeah, so let's look a little more at genetic testing for FH and Lp(a) in Copenhagen, or rather in the eastern part of Denmark. I do it in that part of Denmark, and they are tested on referral, of course, all the patients, and we do NGS, next generation sequencing, and you'll already see some of the well-known genes here, the LDL receptor, ApoB, PCSK9, and a number of other genes. And then it's actually required for referral that plasma Lp(a) has been measured on all the patients, and that almost always happens, not always. And then we answer back to the ordering doctor. We use the American Society of Human Genetics criteria, as you can see here, for the mutations. And then if Lp(a) is measured, and it's above 50 milligrams per deciliter, that's 125 nanomoles per liter, then we tell them what is the percentile for this patient, and now we have actually implemented a figure of the distribution of Lp(a), and we mark where the patient is placed, and we give the relative risk with that level. If Lp(a) is not measured, we ask them please to measure Lp(a), and we also provide an explanation why they should measure it.
So thanks to many people that contributed to our research, PhD students, postdocs. We have a lot of co-supervisors for our students, many collaborators worldwide. The Danish taxpayer, they pay for all of it. Basically, we are socialistic, some would say communistic country, but that's how we fund stuff in Denmark. And there's also a few external funders. And then really important.
Yeah, so many things. We are really, really grateful and honored that you thought of us. Thanks to Catherine, and thanks to all of you who are involved in this fantastic Family Heart Foundation. Thank you so much.
Yeah.
That was great. Thank you.
Thank you so
much. Why don't you guys
much.
sit next to each other in the center, and Josh and I will sit on each side.
Want to come one more over?
Okay.
That's good. Yeah, that's
Catherine
great.
likes a balanced stage.
Yeah.
So we're going to have a little fireside chat where we're going to ask you a few questions, and we'll just have a good chat.
And we don't know the answer, we ask you back.
What do you think?
That was a terrific talk and a great summary of the amazing work you've done. And I'll just say I've always admired your looking at big data, really, which served as sort of an inspiration for the Family Heart's emphasis on amassing this Family Heart database that we'll hear more about tomorrow. You talked about implementation at the end, so maybe I'll just start by going back to Ron's presentation and asking you, what do you see as the biggest challenges within Denmark in terms of actually implementing broad-scale screening, appropriate diagnosis, and then appropriately getting folks on therapy? Like, of the different steps that Ron talked about, where do you think the biggest challenges are?
Maybe I can start because I've been involved in setting up that quality control database. But I think that in itself helps a lot. Because if the different parts of Denmark don't follow the path we put there, they will get this report back once a year. And the directors of the different hospitals, they're used to these reports because there's 80 of them. And if they get these reports back and they're not following what the plan is, they have problems, and they have to go and talk with that cardiology department or endocrinology and say, why are you not doing so? So the only thing that I can see was that we have to work on, and we have a plan to doing that also, actually to make sure that all the GPs refer the patients to lipid clinics. They all have to be seen in lipid clinics. So the thing is to get that referral working better. We're working on that also, to engage all the GPs to send their patients.
Are there enough lipidologists to handle all that?
Yeah, I think so.
Okay.
I think that's quite, I mean, Denmark is a small country, but there's like between 10 and 15 lipid clinics.
Yeah, 11 to be.
It's okay. Of course, somebody discuss about funding and things, but I think if they get these bad reports back, then the hospital directors have simply to fund it. You know, it's tax money. So it's about where do they put the money? Do they put it for cancer? Do they put it for lipidology, or where do they do it?
And do people want to be screened? Like when they're told we're going to screen you do genetic testing, how much pushback is there from people of, I don't really think I want my genes being looked at?
Not much, not much at all. I think, you know, if they are sensibly counseled, not much. People are generally pretty, but what is really lacking, and I think we're going to have to visualize that to people, that is cascade screening. The whole idea about cascade screening is, of course, that you want to get the relatives screened, and for this to be cost effective, so to speak, you need to screen at least 2 per family, and we're not succeeding there. And I think one of the reasons we're not succeeding, we heard about earlier on today, that is that we also have this system where the proband has to be the one who asks the relatives to please go to the doctor, right, because of this problem. And that does not work. It's also not fair, I think. So
to put it all on them. I hear you.
That's simply, and you know, if you have an aunt you don't like, then of course you don't tell her about it, right? Maybe.
But there are lots of other, it's a lot of responsibility to have the proband.
I think it is.
So can I add one thing? So like the genetic testing that Anne is running for half of Denmark, it's paid by the government. Because of that quality control, they have to pay it. So there's no fee for the single patient. It's free of charge. Of course, we pay it through taxes, but the individual patient doesn't have to pay for it. So it's, Denmark's expensive to run, right? We pay a lot of tax in Denmark.
But people are happy there, Josh?
I really enjoyed that talk. I really have enjoyed following your careers, because you always seem to do something that I wish I had thought of, before. I mean, a few years ago, you showed those data on patients not taking statins after negative news reports, just out of the box thinking. But I'm going to ask a more specific question about LPA. You know, it's been 60 years since LPA was discovered. And I'm in a lab in the physical space where Dick Lawn was. There's like still pipettes that have his name on them or whatever. But why do we still understand so little about it? Why do we, you know, what does it do? Why did it involve an evolution just in, and these are not answers that we've had for 60 years, but we're going to have them now in 5 minutes. So why do we have it? Why is it that it causes aortic valve stenosis or aortic valve cirrhosis when other things don't? I mean, what do you think is going on? Was it protective? What is the speculation? What is your thoughts?
I mean, I love speculation, so I'm happy to do that. So you can't trust what I say, but I mean, that's what's in my mind. So Lp(a) developed twice in evolution, the European hedgehog, and in some monkeys, apes, and humans. There's 2 different versions. So it must do something good. Otherwise, it would not be there. As apolitole has homology with plasminogen, and plasminogen is the thing that's important for dissolving blood clots. So when you have a blood clot, plasminogen will help. So you could think that it could, by Lp(a) is very similar to plasminogen, but it doesn't have this ability to stop blood clots. So you can think if you have a blood clot, Lp(a) may be better at stopping bleeding. And certainly in early life, at childbirth, that's where, without modern medicine, many women and children will die from bleeding. So if you have high Lp(a), maybe you survive. Maybe it's a survival advantage. And it seems to me that if that is the thing that, from an evolutionary point of view, makes it be there, then now today we have modern medicine, so we don't die from bleeding at childbirth anymore. But then if you live to old age, so like my age, so, well, are you a bit younger? You look a long.
Oh, I don't know about that.
You know. Anyway, so then being good at stopping bleeding, meaning that you get blood clots. So when you get to old age, that becomes a problem, and then maybe you get myocardial infarction or arterial stenosis, actually, because you get small blood clots, and then it stenos a lot, and that could be the same mechanism for the aortic valve stenosis. Small blood clots that sort of like keep sticking there, and then eventually you get a stenosis. That's my thinking, but it's pure speculation.
So now we have it right now that we're done. I think we know the answer.
I guess what he's trying to say is that it's not just the cholesterol, right?
Exactly, yeah. Go ahead, Dan.
You're possibly the world's leading proponent of the idea that triglyceride-rich lipoproteins are directly causally atherogenic. Could you clarify for this audience, like, what is your view of the actual, is it the triglycerides themselves? Is it the particles that are being carried by triglycerides? What is the basis of that direct causal atherogenicity of triglyceride-rich lipoproteins?
Yeah, certainly, if you take atherosclerotic plaques and you take out the intima and you measure what is actually the mass of the atherosclerotic plaque, it is cholesterol. So that's what's getting deposited in atherosclerotic plaques in the intima. So that's important. And why should the remnants, or people call them the triglyceride lipoproteins, I actually call them triglycerides and cholesterol rich, because they carry more cholesterol than an LDL particles. Mm-hmm. So when they come into the arterial wall and get stuck just like LDL, of course they can cause atherosclerosis just like LDL. And maybe because they're even bigger, they have more difficult to come out again. Everybody would know that, but if, so like from the lumen of the plasma, when you come into the wall, they get stuck there because they can't come further into the arterial wall because of the elastic lamina. So they simply, unless they come back into the lumen again. So I think so. Then the triglyceride part in them could also be important because lipoprotein lipase will degrade it to free fatty acids that are very toxic to tissue. So these remnants, they could maybe also make the plaque more vulnerable to plaque because of local inflammation. Mm-hmm. That's my
I think there's some data even that would support that.
Oh, sure.
Right? That increased inflammation.
Yeah, so I mean, when I joke about it and say it's all in my mind, of course, most of the thing I can defend by scientific evidence, but it's too complicated to explain all that.
Josh?
I like the idea of picking up on what you said earlier, Dan. What else? You know, we had this great talk earlier by Professor Adner. What else? What is the most frustrating thing that you see for LPA, for FH? What are the magic wands that we need to wave as a community, you know, to try to overcome those barriers? I know Dave talked a bit about Denmark, but you know, you have your eyes on the world, you know, like few others.
Yeah, if we talk about LPA, right, first of all, you have to get people to measure LPA, right? And one of the ways we try to start that is that we simply incorporated LPA in the standard lipid panel for all patients who came in with ischemic heart disease and all FH, all patients referred for FH also.
Mm-hmm. Mm-hmm.
I mean, certainly one thing, what we miss right now in these different boxes is the positive phase 3 trials, that if you have patients with high LP-little and high risk and they get it lowered by efficient drugs without side effects, that this will reduce cardiovascular risk. This is one chain that we really miss now. Given that they come out positive, we understood from, I think, that wonderful talk also, this is not the same as it will be a seller, that it will be used. So then I think there needs to be some understanding or urgency among patients that it is actually important to have LP-little, and that will only be by we as a community and many patient advocacy tell about it in media, at meetings, and wherever you can, so people are ready for whenever it comes. And also payers should be available. You can say sort of like a simple thing that just the big success of pharma drug right now is the GLP-1 inhibitors, where if you take those, you lose weight. This is simple to sell, right? It's a bit like, it's simple, because a lot of people know they have overweight, and if they can get this and they can lose weight without side effect, they would even pay for it themselves, right? So we need that understanding among people and politicians and payers that there's a really need for LP-little lowering. So that's what we are here for, and that's what we should all contribute to.
But one of the arguments against it, as you also have heard earlier today, is that, you know, the doctors, they say, but why should we measure it, right? Because what can you do about it? So hopefully we'll have
But they don't have that argument about FH, and yet we still have, you've written a lot about the frustrating problems about FH. So put your hat on for FH and answer sort of the same question. What do you think is the biggest frustrating thing for you, you know, in your care for patients, or the things that you see worldwide that really grates you?
But FH now, so in our country, I know there's a lot of countries where they do, but like in Denmark, like I had this series of meetings with, we have a lot of, like in the US, you have 2 political parties. We have 10 in Denmark in the House of Parliament. But I talk with, so like the main politician being responsible for healthcare in each of them, and discussed this with FH. And when I told to them that, well, this is FH, it's 1 in 200, 1 in 250.
Exactly.
It's a genetic condition. It's extremely simple to diagnose, and it's extremely simple to treat, and it doesn't cost much. Then all of them said, sounds good.
Yeah.
Let's do something about it, and that's why we convinced them. We had a hearing in the Danish Parliament, and they said, okay, here's some money, get this quality control database going. That's it.
I just want to say, I really like the way you've taken your body of research and really taken it to the policy level and to really tried to turn it into action at the policy level. I think that's so important and a lesson for all of us. I think we're going to have to wrap up. I am going to ask one more question.
Yes.
That's okay, Josh, and then we'll wrap up. What do you talk about at the dinner table at night?
How about ApoE or triglyceride-rich proteins?
I think our children, when they were so small, they complain a lot about ApoE at one stage. But we do enjoy, actually, Anne and I, we enjoy life together, and we enjoy whatever we do outside work. So we travel all over the world. We enjoy science, we enjoy music, whatever. Big garden, roses.
I really have always admired your scientific partnership and your life partnership. I just think it's a fabulous thing, and it's been great for the community. So thank you so much for.
Thank you so much.
Thank you.